Thank you very much for this wonderful talk. Now it's my pleasure to invite Dr. Iván Márquez, an oncologist from Hospital Gregorio Marañón in Madrid. I'm very pleased to meet you. And he's going to speak about new drugs for melanoma. Thank you.
Okay, thank you very much. I would like to thank first, especially Álvaro and Anabel for inviting me to this meeting to talk about melanoma from the adult oncologist perspective. And I would like that the advances we are going to present someday, sooner than later, could be translated into these young adolescents and children with melanoma. I want to start with this sentence that it is maybe the most frequent quote you are going to find in any melanoma paper. And it is because, despite it is true, I will try to convince you that recent advances are going to give some hope to these patients. Melanoma has somehow a very polarized epidemiology. On one hand, it is not a very frequent tumor in comparison with another more frequent tumor in adults like breast or colon cancer, and it is highly curable, especially in early stages. But it is the malignant tumor with the faster increase in incidence, and it is, as we have discussed before in the previous lectures, maybe because more concern and overdiagnosis, but it is only in part true, because the rate of thick melanoma and melanoma thickness is the worst prognosis factor in localized melanoma. These thick melanoma rates remain stable, and this is not good news. And in addition, this is the second leading cause of life years lost because of cancer, only after leukemia. And for many years, melanoma patients with advanced stages have a clear standard of treatment, and DTIC has been the only approved treatment for these patients. This could be explained because melanoma is the paradigm of a multi-resistant tumor to any treatment given. And it is because melanoma has three main capabilities in order to overcome the treatment. One of them is an intrinsic apoptotic resistance. The other is that melanoma is capable of edit the microenvironment in its favor. And the other is that the melanoma is the clear example of an immune escape tumor. But fortunately, recent advances in these two features, apoptosis resistant and immune escape system, has led to a breakthrough in the treatment of melanoma patients. The first one is trying to overcome the resistance of apoptosis, and the most important findings have been through the MAP kinase pathway inhibition. Melanoma harbors thousands of mutations, but the most important ones are located in the MAP kinase pathway that is very relevant in many tumors, and especially in the mutation of the BRAF that accounts for almost 50% of melanoma. These mutations leads to a benefit in survival and a benefit in proliferation in melanoma, in metastatic melanoma. And this is quite an exciting example of how basic research can be translated in a real benefit in a very short time to patients. First, there was an observation that nearly 50% of melanoma harbor mutations in BRAF oncogene. Then it was demonstrated that silencing this gene leads to a reversal in the melanoma proliferation. And the good news were that it could be pharmacologically easily blocked. And very promising results were seen in preclinical models in mice in terms of tumor shrinkage and in terms of this animal survival. And they were so promising that in the same year that this molecule, formerly known as PLX, now known as vemurafenib, jumped into the clinical development in a proof of concept of a phase 1 clinical trial that treated with an oral inhibitor of vemurafenib patients, multimetastatic, multi-treated, that had a melanoma with a BRAF mutation. And they observed not only what we want to observe in phase 1 clinical trials, that the drug is safe, but also a spectacular, really unseen responses for these patients, as you can see in this slide. And in the next year, it jumps to the real deal in clinical development, that is a phase 3 clinical trial that confronted the so-called standard DTIC versus vemurafenib in patients with non-treated before with stage 4 melanoma. Stage 4 was better in first and second line in terms of progression-free survival and also in overall survival than classical chemotherapy. So we have 3 drugs, 3 studies that demonstrate that in BRAF mutant metastatic melanoma patients, it is better to treat with this treatment than with the so-called before standard treatment of chemotherapy. And these treatments are fairly well tolerated. There are almost no grade 3 or 4 adverse events, but we have to be watchful and we have to monitor this treatment because sometimes patients can experience skin toxicity with very rare events in form of secondary tumors, hepatotoxicity, pyrexia, and in the case of trametinib, heart and ophthalmological toxicity. But again, so far, so better tolerated than classical chemotherapy. The other breakthrough is overcoming the immune escape, and this has came thanks to the studies in immunocheckpoints. The study of using the immune system against the tumors has been somehow a long story of fails, maybe because we do not fully understand how inflammation can be both a cause of cancer and a part of the solution. And in that way, for many years, immunotherapy, not only in melanoma, but also in many solid tumors, has failed, retrieving very, very poor results. But this was changed in maybe the magical year for melanoma, 2010, when all the breakthroughs came. And this was because researchers realized that using the immune system was not somehow triggering the accelerator, but releasing the brake of this immune system. And it was the proof of concept started with 2 different experiments, one led with to Alan Sharp and the other by Jem Allison, that demonstrated that this molecule, CTLA4, deficient mice died because a massive infiltration of T-cell activated lymphocytes. So the CTLA4 naturally play a role as a negative regulator of the immune system. And in the other way, this could be pharmacologically inhibited as form of antibodies blocking CTLA4, demonstrating regression in preclinical models of tumors. And that started by the end of the millennium with Medares when it started to develop ipilimumab. In order to understand ipilimumab, we have to understand how CTLA4 works. When there is a presenting cell showing the antigen to the T cell, there is a very weak activation signal. So they need a secondary signal that leads to an activation and proliferation of T cells. But in order to not create an autoimmune response that could lead to sickness, naturally, lymphocytes are counter-regulated by CTLA4 that sends some kind of a negative signal. So if we block this negative signal, we are releasing the brake. We are making that T cells could remain active. And this theoretical concept was demonstrated clinically with early clinical trials that, on one side, demonstrated clinical responses with these antibodies, and the other side, autoimmune side effects. So this is demonstrated that CTLA4 inhibition could work this way. But maybe the most important observation in the field of immunocheckpoints was that a really significant percentage of patients treated in different trials with ipilimumab had a long-term survival, never seen in melanoma. And this even changed the objectives of the initial clinical trials in order to look at overall survival instead of responses. And here it comes, the year 2010, when for the first time in a phase 3 clinical trial, something demonstrated an improvement in overall survival in patients pre-treated with melanoma. In this clinical trial, ipilimumab was compared with an experimental vaccine, and it demonstrated in patients pre-treated with another drugs an improvement in overall survival. But maybe this is not the most striking finding. The most striking finding is that a high proportion of patients survive beyond the 1 year, and beyond second year, a quarter of patients are still alive. This in melanoma is really striking and never seen. And the fact is that these long-term survivors remain stable in time. It was also demonstrated in the first-line setting in combination with dacarbazine. Also, it was demonstrated these long-term survival rates. And to be more optimistic, all the patients treated both in clinical trials, both in expanded access, when they achieve the 3-year survival, the cures reach somehow a plateau, and we can talk about patients with metastatic melanoma that otherwise will have a very poor prognosis. Some of them have an unstable disease for even more than 10 years. But not everything is easy with ipilimumab. There are two main challenges. We have to assume that we have to look at the new responses patterns, that we have classical responses pattern of patients that respond, patients that are stabilized, but some patients experiment at the beginning somehow a progressive disease, that it is all the contrary. It is the first manifestation of a ulterior response. And it is quite difficult because we do not have any tool today to decide which patient is really progressing or which patient is responding. And the other barrier is the toxicity of these treatments. Due to the autoimmunity that they provoke, nearly every system or every organ can be affected, especially skin, liver, gastrointestinal tract, endocrine, or neurological. But fortunately, most of the side effects are very mild and can be preventable or even treatable with the steroids use. So, with this in mind, this is what we have today in Spain. Today in Spain, beyond dacarbazine that was the past, in the BRAF mutated patient, we can use BRAF inhibitors. For regulatory issues, we still in the wild-type patients have to use chemotherapy, but wherever the patient comes after the progression, ipilimumab can be a very good option. But this is, and I have to say that happily this is not still clear, because despite ipilimumab, dabrafenib, trametinib, or bemurafenib improve survival, there are still barriers we have to overcome. And the first barrier is that these treatments have very different patterns of clinical utility. While ipilimumab has long-lasting survivors, there are few and slow responders, and we have not any predictive marker in order to decide which patient treat with ipilimumab. On the other hand, bemurafenib and dabrafenib have many and deep and fast responders. We have a very useful predictive marker, that is the BRAF mutated status, but unfortunately, most of these responses are short-term. So it comes the question, we have a very powerful treatment with a fast and deep responses, and we have a very durable treatment that makes long survival. So why not combining them? But unfortunately, this logical idea failed because it retrieves unacceptable toxicity in terms of the hepatotoxicity. So finally, they didn't get well. And here is where they comes the very new treatment. We have talked about new treatments. We have to talk about the very new treatments that are going to try to improve these responses in two different ways. The first way is try to overcome BRAF inhibitor resistance, and the other way is to explore new immunocheckpoints strategies. The BRAF inhibitor resistance is a big problem because we have patients that respond fast, respond well, but in many cases they have a progressive disease sooner than we would like to. And it is due to many molecular mechanisms that we are not going to discuss. They can be primary, they can be secondary, and there is a huge effort in order to try to overcome this. It has been explored, different rhythms, different sequences, retreatments, but the clue and the most interesting data are the combination of the BRAF inhibition and MEK inhibition. And this clinical trial, that it is a phase 2 clinical trial, compared the combination of dabrafenib and trametinib versus dabrafenib alone, and they realized that their response rate was doubled, and also the progression-free survival was increasing more than 50%. So this is very exciting because the primary resistance are overcome with this combination. But we have to wait to the phase 3 clinical trials that are currently under analysis. And one bonus track of this treatment is that this combination prevents skin toxicity, especially skin tumors. The new immunocheckpoint strategies are because there is not only CTLA-4 in this negative regulatory T cell function. There is a player, there is a huge number of immunocheckpoint regulators, and the most advanced one is the PD-L1, PD-1 immunocheckpoint. One of the first used in a phase 1 clinical trial was lambrolizumab, that it is an anti-PD-1 antibody. And with this antibody, researchers observed a high rate of responses, high rate if we think in immunotherapy, also in ipilimumab patient, and seeing these graphs and these CT scan images, it reminds us something that we have seen before. It reminds us the effects of the BRAF inhibitors, these spectacular responses, but with one big and important difference: patients that still have these responses even without treatment and that have long-time survival. These results are very similar with another PD-1 inhibitor, nivolumab, that was published recently. But the most anticipated and the most exciting way of these strategies came from the combination of the ipilimumab and nivolumab through the results of a very early phase 1 clinical trial that tries to assess what is the role of this combination. In this clinical trial, again with very few patients to make solid conclusions, they observed that not only there were many responders for immunotherapy, but that the responses were fast, deep, and long-lasting. So this is really exciting to see how the former treatments can be overcome for this. But again, these 3 clinical trials are phase 3 clinical trials, phase 1, and we have to wait to phase 3 clinical trials. So I wanted to repeat this slide because if we are lucky, this slide is going to be old-fashioned in a near future. And it is going to be old-fashioned because all the new treatments with the MAP kinase inhibitor and other kinase pathways inhibitors, new immunotherapy treatment that rather than from being a problem is really an opportunity to offer to our patients. And the slide that I wanted to do for my conclusion is that this is somehow a happily problem, and how to handling this problem is very easy. It's just to keep calm and carry on researching. So thank you very much.
So far, the only approved treatment for melanoma is interferon. So are you still using it, or are you really not recommending interferon for your patients? And which are your recommendations, personally ones?
As I said before, all the decision on therapy we take in the Italian group is led by the adult group. So we ask them what we want to do. And I don't know why, but the Italian group on melanoma is against the use of interferon. To be honest, I never use interferon in our patients, and know any patients in the TREP series use interferon.
Okay, in adult oncology, interferon somehow is the new DDIC. It is a treatment approved in 1996. His use is approved for adjuvant treatment in high-risk melanoma, but it hasn't, unfortunately, demonstrated an improvement in overall survival, or only in progression-free survival. All the strategies that have trying to substitute interferon have failed in that way, because only have a progression-free survival benefit in comparison with observation. There are clinical trials that are trying to explore these new treatments in the adjuvant setting, vemurafenib versus observation, and ipilimumab versus observation or versus interferon. We are using interferon in high-risk melanoma outside clinical trials because it is labeled, and you have to offer to the patient, but you have to discuss with him or her all the secondary profile, effects profile that it is really hard to wear, and the possible benefits in terms of progression-free survival, not overall survival. But we are still using it.
Okay, fantastic. Any other questions? Not anymore? Yeah, he has a question. Wonderful.
No, it's half a comment and half a question from them, or for the group, because I think that the major problem is how many patients in the adolescent young adult group have the access to the new drugs. And it's a big problem because the two clinical trials I talked about, the pediatric trial with ipilimumab, the pediatric trial on vemurafenib, are not going so well. There is a very small number of patients registered, less than the expected number. And the reason is that it's difficult to build this type of trial, because the trials are led by the company, and the company decide what are the centers that are contacted and enter in the study. For example, in Italy, we have 3 centers, but one center is the center of Milano that leading the melanoma group, but the 2 other centers are the 2 centers that never treat any children with melanoma also in our series. So there is no any reason to ask them to enter. While, for example, the center of Padova, that has many patients with melanoma, are not called to enter. So I think that our task may be to build the structure that may be half the company and the national group, for example, to try to find a way to help the company to find the right center, to help the TCC, for example, to contact the right person. And the other point is that our group could be that the structure that can let the company understand that, okay, they have to do the pediatric plan, so they want to have a pediatric center, but in this type of tumor, they need to work also with the adult centers, because the majority of the pediatric patients are seen by adult centers, and it is impossible that an adolescent treated in an adult center will be sent to pediatric center when they have metastatic relapse. It's never happened. We have the same example with GIST. No patients enrolled in the GIST protocol wild type in Italy because there are only pediatric units and not adult centers. So we have to do this job.
I quite agree. It's a complex mixture of different things that are happening. Some of those things are about the slightly different agenda for the pharmaceutical industry in obtaining approvals for its agents from the clinical agenda. Some of it is about the behavior of different hospitals and organizations and individual clinicians about sending patients from hospital to hospital or place to place. Some of it is simply about knowledge and understanding what options there are and what is available. We can and indeed we must, within our future work, provide an interface for studies in the key adolescent and young adult tumors where people can come to us for advice how to deal with these problems. And that is clearly already the case in the United Kingdom, that the existing structures serve that function. We can also try, although it will take some time, I'm sure, to influence the grant-giving bodies for academically driven trials and the regulatory bodies for industry-driven trials to consider the epidemiology of the disease when they design the age criteria for the study. You probably know that in the United Kingdom there was a terrible situation as far as this was concerned about 10 years ago, and trials were either written by children's groups with an upper age limit of 16 or perhaps 18, but usually 16, or adult groups with an upper age limit of 18. If you were 16 to 18, sometimes you couldn't get into any of the open studies when you got these diseases. Sometimes there were two very similar studies being devised by similar groups that hadn't communicated with each other and duplicating the required resource. So we worked with our regulators and with our charities to put a box on the form when you submitted a trial to the funding organization that said, how do your age entry criteria fit the epidemiology of the illness that you're wanting to study? And this resulted in people very comfortably over time changing those criteria. And we'd like to work with perhaps the European Medicines Agency, certainly the EORTC, certainly other children's groups such as the ITCC, to continue to achieve these different changes in different ways. A network of individuals who know what else is available, a resource for people trying to navigate these things who don't yet understand how best to try to do that, and some work with regulators and funders to try to encourage logical trial design that is only fair to teenagers and young adults. It doesn't have to advantage them. It just has to be fair to them compared with younger children or older adults. I hope that's helpful.
Okay, thank you very much. An announcement from Alvaro. Thank you very much.